All right, so my name is Vicky, and I'm here to talk to you about the effects of gear type and season on Vibrios after routine handling. So the interesting thing about off-bottom aquaculture is that y'all have access to your oysters at all times, and so you can do these routine handling practices like desiccation, tumbling, sorting, and grading. That way you can prevent barnacles, overset, mudworm infestations, all of that stuff. And overall, the goal of doing all of this is to improve your oyster quality and earn that higher profit. But when those oysters are removed from the water, they are not filtering, they're closed up, and they are exposed to higher air temperatures. So during that time, that creates a perfect little incubator for Vibrio bacteria to grow. And so routine handling does cause increased Vibrio levels. For those of you that don't know what Vibrio are, they are pathogenic bacteria, and they're naturally found in the waters where your oysters grow. There are two species that are a public health risk. The first is Vibrio vulnificus, or VV. This species is important because it has the highest case fatality rate of any foodborne pathogen out there. The second species is Vibrio perihemolyticus, or VP. This species is important because it's the leading cause of seafood-related infection in our country. So now that I've scared y'all against eating oysters, I'm going to tell you that farmers can put their oysters back in the water to fix this problem. So once you re-submerge your oysters, they're happy again, they'll open up, they'll start filtering, and that allows those elevated bacteria levels to go back down to ambient levels before you harvest. Now, currently in Alabama, we have two different re-submersion requirements. Depending on the gear type you use, it's either 7 or 14 days, and I'll explain more. So in the past in Alabama, we've done some studies to look at if there are differences between different types of handling and what that does to the re-submersion period required. So the first study was done in 2014 and 2015, and this looked at simply air drying your oysters. And then we moved on and looked at a rougher handling type in 2016 and 2017, looking at different tumbling and refrigeration combinations. Now, both of these studies were only performed during the summer months under the assumption that the summer has the highest risk for Vibrio infection. And again, these studies were also only performed on one gear type, the adjustable longline system. But regardless of that, they produced consistent results that a 7-day re-submersion period was sufficient regardless of the handling type. And this actually allowed us to achieve a reduction in the re-submersion period from 14 to 7 days, but with the caveat that it could only be for adjustable longline system farmers. So with that, we had some other questions about what could affect re-submersion time. And so the first question was season. We only looked at these things during the summer months, but different times of the year have different variations in water and air temperatures, like in the spring when the water is colder but the air temperature is heating up. So could that affect the re-submersion time? Our next question was a question of gear type. Like I said, we got that reduction, but only for farmers on one type of gear. So the issue with gear type is that they hold the oysters at different positions in the water column, which could affect the re-submersion time and Vibrio within the oysters. So in the picture on the top, you see the adjustable longline system. This system will hold the oysters at a fixed height above the substrate or above the sediment, and then the water level will actually change relative to the oysters based on the tides, versus the floating oyster grow system in the picture on the bottom, where those cages are floating. So they're actually at a fixed depth below the surface of the water, and they'll float up and down with the tide. So these gear types and where the oysters are located in the water column could affect the re-submersion period as well. And then finally, we kind of wanted to expand outside of Portersville Bay, which is where the two studies were performed previously. So we had this grand question of, is it different on the East Coast versus the Gulf Coast? And so we performed sister studies in Cedar Island, North Carolina, and Grand Bay, Alabama. But for the purpose of today, I'm just going to talk about the season and gear type effects that we looked at in Alabama. So for this part of the study, we used Auburn University's research farm in Grand Bay, Alabama to perform two trials. One was done in early May to capture that springtime, and another in the summer month of July. Now for this study, I looked at both of the gear types side by side, the oyster grows and the adjustable longline system. For each gear type, for each gear type, I had 3 treatments. The first is a submerged control treatment. These oysters never left the water. This lets us monitor the natural levels of Vibrio within the oysters during the experiment. Then I had a desiccated treatment where those oysters were air dried for 24 hours and then put back in the water. And finally, I had a tumbled and refrigerated treatment. These oysters were removed from the water, sent through a tumbler once, and then placed into a walk-in cooler overnight for 18 hours, and then put back into the water. Now, over this 2-week trial, I wanted to collect samples at several time points. That way we can look at how those Vibrio levels change over time. So first, I collected samples before I did anything to the oysters to get a baseline. Then I had the treatment period where I removed the oysters from the water and applied those treatments. Then, immediately after those treatments were applied, right before I put them back in the water, I collected samples and then put the oysters back in the water. Then, during the 14 days of resubmersion, I collected samples at 1, 3, 7, and 14 days, took them back to the lab, and tested them for Vibrio. That way we can determine when those levels went back down to natural levels within the oysters. So each sample was tested for 4 different Vibrio types. We had total VV, total VP, and then the pathogenic VP strains. So today, just a few preliminary results from last year. I know all of y'all like to look at the environmental data, so I can show you that our salinities between the 2 trials were similar, but we did capture a much cooler water temperature in the spring month, which is exactly what we were aiming for. So all the graphs are going to look the same, so I'll set them up. This is going to show exactly what happened after those treatments were applied in July. On the horizontal axis, you'll see the 4 different types of Vibrio that we tested for. On the vertical axis, you see log NPN per gram. That is the most probable number of bacteria per gram of oyster meat. Now, for each gear type, the green bars are the submerged control, the blue bars were the tumbled and refrigerated treatments, and the orange bars are the desiccated treatments. So you see, immediately after those treatments were applied, we had a 1 to 2 log increase in Vibrio bacteria in those desiccated treatments. And surprisingly enough, after the refrigerated treatments were taken out of the walk-in cooler, they had no significant increase in Vibrio bacteria. What's interesting to look at is the comparison between the gear types and the submerged control oysters, and we actually found no difference in the Vibrio levels across all the types between the 2 gears. Now this graph shows after 7 days of resubmersion in July, all of the Vibrio levels returned back to ambient levels, except for one exception in those tumbled and refrigerated treatments. They did not recover with respect to total VP levels until day 14. Again, I just want to point out that we did not see any differences between the gear types in any of the treatments in Vibrio levels. All right, so now I want to talk about what happened in May. So again, this is the cooler time of the year. And you can see that immediately after those treatments were applied, we still had the same 1 to 2 log increase. So while that air temperature is warming up, even though it's cooler in the water, you still get that 1 to 2 log spike in Vibrio levels. While the refrigerated treatments had no significant increase in Vibrio levels, we did not see any differences in Vibrio types between the submerged controls of the 2 gear types, except for total VP, where we actually found that the adjustable longline system had higher levels than the oyster grows did. But what's interesting to note is you think summer months have the worst case scenario for VP risk, but actually we found higher pathogenic levels in all of the treatments in May than we did in July. So again, this is going to show after 7 days of resubmersion in May, we found that all of the Vibrio types recovered back to ambient levels. And again, just noting there are no differences between the 2 gear types in Vibrio levels. So in conclusion, we found that 7-day resubmersion period has a wider applicability with this preliminary data, with that one exception. So regardless of gear type or handling type, the Vibrio recovered after 7 days. We did note that pathogenic VP was higher during the spring month than it was in the summer month, which might indicate that summer months might not be the worst case scenario for VP risk. So in the future, we are going to continue this study. We have another set of trials to perform in 2019. That way we can see if that 7-day trend continues. And then we are also doing a sister study in Cedar Island, North Carolina, using the Oyster growth system. And the experiments are set up that way we can compare the Alabama and the North Carolina data to each other. Additionally, we received funding to continue to chip away at that question of geography. And so we are doing similar re-submersion studies along a smaller geographical scale using neighboring water bodies in Mississippi, Alabama, and Florida. And with that, if I have any questions.
Any questions for Vicki?
Yes, sir. So this is completely separate from that VARB study that was done. This is kind of supplementary to it. So we have to wait until after this next year's data is complete to present it to the correct authorities. So he asked if the salinity levels had any effect on Vibrio. They do. We are working at trying to create an analysis that can incorporate our environmental data into it to see if that's affecting anything. The average temperature was 30 degrees Celsius, or about.
That was the water temperature, though.
The water temperature, but the air temperature was about 90. Not that I noticed, no. But I was using diploid oysters, not triploids.
Thank you, Vicki.